Cette conférence va maintenant être enregistrée.
Okay. So everybody heard me now. And Yann, you hear me?
Yeah.
Okay. So good afternoon, everybody, and thanks to be here for the last webinar of the year. So it's my pleasure today to introduce for the first talk someone you probably know as a former member of the RFMF board for many years and with the driving force behind the workflow for metabolomics, as you know. So it's Yann Guitton, which serves currently as manager of the Melissa Metabolomic Core facility in Nantes, and is involved in many research and development from agronomy to mass spectrometry, and also, as you know, in the development and the maintenance of workflow for metabolomics. So Yann, the floor is yours, or better, the screen is yours for 40 minutes. So to present us so many incredible tools. So let's go for the dream. And
Yes, thank you, Pascal. You're right. It's time for me to talk about dreams, maybe a bit of that. So I'm really happy to be there in front of you all, a lot of people today, and I hope that you're not expecting training lessons on LC-HRMS annotation tools. It's not the purpose of today. It's just the idea behind my talk today is to cover parts of that big word of annotation and using HRMS data today. So nothing is moving. Oh, yes. So just an overview of the today's story. Really simple thing about why we are doing annotation and what is our current road, where do we need some help, and when I speak about help, it's a software to help us to go through the annotation process, and I would try to give you some tips and tricks to go further on the annotation and not to maybe miss part of the path that will allows you to go to your final goal, the annotation. So, as you all know, in fact, the story is quite simple. Our aim is simple. We just want to crash any kind of organism, and we want to characterize and give a name to every single ion that we can see in these organisms. So the game is simple. The rules are also simple. Again, here bacteria, then we do extraction, some kind of chromatography or separation, and then we apply mass spectrometry. And usually today, high resolution mass spectrometry affiliated to liquid chromatography, but can be GC, can be CE. And in the end, we want to have a nice report with molecule names and when we can, we also want to add to that some quantification data. So simple rules, but depending where we start, you all know that graph and with the 5 levels, and so we can start the game at level 1, where when we are doing some quantification with on a known list of standards that we know by heart in the lab. But usually in our untargeted approaches in metabolomic lipidomic, we may start at level 3 with some candidate with a suspect list, and then we try to go to level 2, and the goal to go to level 1 with a confirmed structure and even to a better level. And in our everyday job in the platform, people ask us to do the fully non-targeted screening approach. So when we start with just a M/D and a retention time. And sometimes we have some other information like ion mobility or other UV data, let's say, for that feature. So we are for some time stuck at that level 5. And if manually we can try for every ion to go up to that level 1, it takes us a lot of time and efforts, and that here where we need some softwares to help us to go to all to the end of the pipeline. And so many articles are talking about those levels, and so the game is known from everyone. And sometimes we have some variations in the rules, so new classifications, but the aim is always the same, is to go from just a feature with retention time and M/D exact mass to confirm structure, going through different steps. First, giving the name of the species, elemental formula, and all that. So to be honest, that game is quite an adventure, and sometimes we are starting with just a few tools, and the aim of the today is to give you not a full overview of the existing tools, but maybe some clues on where you can find new tools for your adventure. So usually we start with features, as I said, and slowly we go to statistics. We filter our ions, and then we are happy because we found some really nice biomarkers that explain everything in our organism, and at least that give us some really interesting molecules to annotate. But when we start the annotation, in fact, we sometimes start a really difficult part of the workflow, to the main bottleneck still today. And it's take us sometimes to go to the final goal site, to give a name, to identify, not just annotate, but identify the biomarkers, to give a name and confirm that with a chemical standard. So to do that, to go to really the end, the main goal, sorry, of that, to the level 1 tracer, we need some help. We need some help from software. We have a lot of software packages that can help us to give us some clues, maybe not just the right answer right out of the click of a button. So that's one of the take-home message of today. These tools are wonderful. These tools are more and more user-friendly. They give us they're faster than before. They give us a lot of information, and give us putative annotations, but these are just clues and not full proof of the correct identification of our features. So learn how to use these tools, but keep your mind open and have some critical view on the results before jumping to the end of the game. So very good tool, very efficient tools are present on I would say that's not on the market, because most of the tools that I will present today are open source and free tools. We have also really good tools from our vendors. And the story starts at different steps of the workflow, because we have some tools and software that can help us to have good annotations that start at the really beginning of our journey. So some few tips and tricks at the time of the acquisition can save you a lot of time at the annotation step. Again, when we have some tools that can filter out the data and help us a lot to improve our annotation process. Then for structural elucidation, we have new tools available that are easy to use and that can give us a lot of useful information on everything. And in the end, when we are happy with the annotation process, we can share these results in a ordered way and help the community to learn a lot based on our good annotation process. So many tools. So just here a few screenshots from a GitHub repository from Marshall Spicer, and where a list of tools have been created some years ago. And if you go in the list, you see a lot of tools. Every year or so, we have some good reviews in journals about all the resources and tools that are available for the annotation part of our metabolomic and targeted studies. And we have also other repository, and I love the meta-herbolomic toolbox. That is a GitHub page where lots of tools are described and categorized for every step of the metabolomic process. And in that metabolic workflow, we have that annotation steps, and a lot of tools are really finely described and are accessible at just a click. So I love interactive sessions. So now it's your turn to speak, and at least to react and tell me more about your everyday tools, because I was saying that it's difficult to speak about all the wonderful LC-HRMS annotation tools. So if you can go on wooklab.com, and it's websites, and you can then enter the event code, which is RFMF webinar. You will be asked to connect, so you just can connect anonymously, and then I will launch the questionnaire. It's 2 small questions, and I really want to hear from you. So I hope it will work. It's first try, so let's go to wooklab.com, enter the event code RFMF webinar, and connect. So let's start. Hope it works. Go. Okay, so now if you click on an answer, yeah, thanks. People are answering. Yeah, 60 people answered. So half the quorum. I'm curious, I want to know more. Please Yes. Almost 80 people, perfect. Thank you so much for your reactivity. Still people answering. I don't want to spend too much time on that, so just a few sec for the last participant. Oh, let's go to 100. 5 more. 4. Okay, I would start to. So we have a winner, let's say. So we are almost equality. So MZ9 is first, R packages second, and then vendor stamp were MS dial and some others. So so we have W4M, twice vendor, and own in-house algorithm. So yes, that's good to have to see that Python tools. And on yes, W4M and MZ9. Okay, I don't know. Scripts, okay. That's a good thing. Progenesis QI for the vendors, compound discover, metaboscapes, depending on yes, MSDAL because user friendly. Okay, well known, perfect, great. Thank you for that. So this was the first WOOC Labs. So stay connected, stay tuned. I would go back to the second one. So second question. Which tool do you use for feature annotation? So the first one was for feature finding. And do not hesitate to add some comments on your answers. Okay, 50 people. Just don't want to spend the whole time on that. It's still good to have your input. So here we have a serious CSI finger ID in front, then vendor software and GNPS. Okay, great. Perfect. A lot of others are coming. Okay, good. So I will just stop there and have a look at the others. For level 1 in-house database, the gold metabolism, because yes, for GCMS tools from Thalassomics, of course, I know you, Samuel. And yes, I agree, these tools are really cool. Scan annotation, yes, a brand new one on the market. Congratulations for the people in Rennes. And yes, workflow like generator like Canime in-house. Okay, a lot of in-house. That's good. Okay, and then my Comanut. Yes, confirmation is standard. No, not a lot of Pythons. Okay. GNPS for sure. MedGem for networks. Okay, great. Thank you all for that. So it's good to see that most of the tools the community is using are in my talk today. Great. I'm happy with that. Okay, so I just want to click, it's not moving again. So a lot of tools, and I would say that annotation can start really early in the process. And I want to talk about one tool called IE Omics. which is made for iterative DDA generation. So you all know about data-dependent acquisition modes, where you are triggering your instruments to capture the top K ion in every spectra and fragment them. So here we have a top 5 example. And so it's a powerful tool, but the trouble is that it's impossible with just one injection to have a full coverage of all the ion present in your extract. And sometimes the triggering of the isolation, the fragmentation, is made at the low level of intensity of ions in the front or the end of a peak, elution peak. So sometimes not that reliable quality of MS2 spectra. And someone came, so Jeremy Commel came some years ago with the idea of iterative DDA runs. So the idea is quite simple. Just run your extract several times and add the capacity of our instruments to have exclusion list in mind when we are doing DDA. It's just an R script, quite very simple to use. So first you run a full DDA run on your sample, usually a QC sample. Then you convert your file to MS2 format using MS Convert, the very same software you use to create MZML files. And you add that file into a really user-friendly scripts. It's in R, but we have pop-ups that you have to fill to say, okay, positive mode, I want to select this and that. So it's really easy to use. It will create an Excel sheet with a list of ion that you can import into your acquisition method, and then you start again a DDA experiment, but now with an exclusion list added to the method. And you start the second acquisition, and when the new spectra come, the system will check if it is allowed to fragment this and these ions. And so slowly and step by step, you can do, let's say, 3-4 injections, consecutive injections of your samples, and have ions with lower and lower intensity selected and fragmented. So you have a really good increase in the number of metabolites of ions that are fragmented with that iterative DDA experiment. So please go and read the papers and take the time to download the R script. It's really easy to use and can give you a lot more information in your data. And these tools are, that script is available in some other way into our vendor softwares, like in the Acquirix, in Thermo Fisher, Agilent with the lipid annotator, use quite the same process where in the injection sequence you have repeated DDA experiments with exclusion list creations. Really recent tools, but based on old tools, the virtual metabolomic mass spectrometer, it's also a way to improve your MSMS acquisition methods virtually. So again, if you take the time and have a look to that VIMS thing, can really help you to improve your acquisition methods without doing a long design of experiments with real sample on your machine. Can be done all in silico. Other tools are really interesting, but I won't go through today. It's Skyline that now have some metabolite features, metabolite annotation features, and lipidomics related modules, add-ons. For those of you who are doing lipidomics please have a look at these. Maybe it can save you a lot of time. So we have seen that in the survey. MS-DIAL is one of the software that you use commonly in your platforms and during your analysis. MZMine was the first one. And we are now seeing others. So I would talk a bit of Deimos. It's a Python package available, made available recently. And it adds the advantage to have good documentation. And this is new because some years ago, we already have really good, nice tools in R, in other languages, but the documentation was not that high quality. But today things are really improving, and we have example data sets, we have example codes to run and everything. So again, and here we have a tool that is able to process ion mobility, and that MZMine can do that also. And we have for sure all these Pythons and R scripts and modules available, but maybe not always that user-friendly. So just to focus on Deimos, so I think that tool is interesting, and it's an example of the new tools built today by the community of developers. So tools dedicated to our users with nice examples, nice tutorials, guidelines, examples, and data sets, test data sets. Nice also visualizations. It's not the only one to do that, and that's a good thing today with our incredible new LC-HRMS acquisition process. And we have that serious CSI finger ID tool that would go a bit further in, and we saw that in the survey that some of you are using it. And why we are using these tools, MZMine, MS-DIAL, and Sirius, it's because they have user interface and intuitive design. It's just a click away. I don't have the time right now to make a new game and to ask you in the chat if you're able to recognize the pictures linked to Sirius, the one linked to MedGem, the tool that allows you to visualize your MSMS networks or MZ9, but I'm fairly sure that most of you are able to do the connections, and the answer is there is. So pretty sure that everything is okay for you on that. And the idea here is to show you that these user interface are really powerful and really intuitive to use. But a bit of warning, powerful tools means also big risks. Why I say that? It's because when you are doing automatically the things, it seems easy. It's still easy to click on a button and obtain a lot of annotation results. But the trouble is that it's also fairly easy to propagate mistakes, misannotation, if we are not taking care of the way the tools are functioning. So please read the manuals and keep your mind clear when you are seeing a result. It may seem so wonderful results, but maybe the first hits in the list, it's not the good one. Keep that in mind. In tools like Sirius, most of you know how to use it, but maybe you don't see or didn't see yet that there are hidden treasures in these tools. And here the arrows show you that you can filter on logP values, the results, so you can add some user inputs in the results and re-rank the results. You can use that. For those of you who are doing want to know more about the link between the proposed structures and your data, you can just click on these squares here. It will highlight the parts of the molecule that is explained by your MSMS spectra and the associated fingerprints. You can also see if the results was found in the training datasets. So it's written here in black. We can see that the first molecule was in the training dataset used by Sirius. When you can have also some insight into the annotation, where you can see in your MSMS spectra which ion is have been explained by the tool. So lots of nice features are here to help you understand the logic behind the proposed annotation. And for those of you who are doing lipidomics, just click on that tick box and it will use El Gordo algorithm to tag lipids. So if you have done some lipid extraction, do not forget to click on that small box. All these tools are actively developed by their teams, and so you can have information on the change logs. You can find out the new release. So regular release means also new options for you. So please have a look at this, and you can maybe improve your workflow and know more about the tool. Regular evolutions can be seen on the GitHub. If the tool is available on GitHub, you can see that these tools have been modified 5 months ago. And now for most of the tools, we have nice articles. And here we have a tool that is able to compare really, really fast our MSMS spectra to big databases. So this was an issue some time ago, the time it takes to compare a big list of MSMS against a big database. And now we have tools available to all of you as a graphical user interface or as a command line that is able to compare really, really fast. It's really easy to use. Just import your MSP file with your observed spectra or your MZML file and the MSP database that you want to use for comparison. And if the database is really big, the first execution will take 10 minutes to half an hour, but then all other execution would be really, really fast. Regular updates means regular options, new options, but sometimes a new version means new troubles. Yes, because some tools, that one I developed, for example, for workflow for metabolomics, use external tools like Sirius. And when Sirius moved from version 4 to version 5, it broke part of our workflows because now we need an account to use it. So we have to go back to the development and correct that issue. But it takes sometimes us a bit too long when we listen to you to correct these small bugs or small changes that are independent. So new versions sometimes have an impact on other tools. Sometimes are not always moving, but for EMX, the tool is mature and it's still functioning. So 5 years with no change means not always mean that the tool is not used anymore. Don't be afraid, and these tools are more and more easy to use and to understand, even if you are not a bioinformatician. Please give them a try, ask around you, how to use them. And I want to focus now on a new tool that's called MassQL for Mass Query Language. It's a tool that allows you to ask questions to your spectras. It's not a magic thing. It's all done with your knowledge on your mass spectras, but with that query language, we are able to process the spectra without picking, and we are able to find in the data region or scans of interest that are, let's say, contains special neutral loss that is of interest to you, or a special isotopic profile, let's say chlorinity profile, or thing like that, with just by just using opening the MZML file and creating a small query. Not every one of us is able to write SQL query like that, but the MassQL magic is that some people already write for us some queries for some really important families, that these queries are already available online. And when you want to try by yourself to build a query, the developers of MassQL have think of something really simple. They create an English or Portuguese or French translation of your SQL query. So here in that example, we see that we are searching in the MS1 level, and we are searching for M/Z of 88 or for something with a M/Z tolerance on 0.1, and everything is explained in plain English right below. So it's help us to will help you to play with that tool. When you are using a tool for the first time, you can try to read the code, but to search for hidden functions. But it's not our main interest as a chemist. We are not bioinformaticians. But what we can do always is when the tool is not functioning or not doing exactly what we think, or we're not understanding something, please do not hesitate to create issues and to ask questions to the developers. It will help us to improve our tools and maybe to guide you through your path in the data analysis. It's also a way to give the developers some bravo. So now it's your turn. It's your turn to start the adventure and to annotate your data. You're not alone. As I was saying, many new ways of teaching how to use the tools, tutorials for Galaxy. We have those Galaxy training networks. You have the YouTube. You have the online documentation in many ways, and a lot of tutorials everywhere. Read them and continue the adventure. Thank you all. And just save the date. If you're in France in Saint-Malo in June, please come to Saint-Malo. And if you want to know about workflow for metabolomic, you can ask us, and see you later in front of your computer or in a congress. Bye.
Thank you, Yann. Thank you very much for your presentation. So we have a question in the chat from Loïc Mervan,
Yes.
I think. Do you want to ask your question, Loïc, or maybe it will be... you don't have a mic. Okay.
So it's
So
in chat.
as you mentioned, DDA can sometimes suffer from low quality. It's okay.
Yes, the connection. Sorry. So yes, Loïc, if we go lower and lower, it's we have a decreased quality of the MSMS. So that's why the tools for annotations now contain some QA/QC verifications.
Ah, donc en fait, lui disait que c'était peut-être plus intéressant de ne pas faire du data dependent, mais de faire du...
And we can also use the data independent analysis and use other software for the deconvolutions. But again, we need some many points to have a nice deconvolution and nice MSMS spectra.
Okay, thank you, Yann. So have you other question for Yann? No. Maybe I have one, a tricky one maybe. You've presented us a great range of data processing software. What starter pack would you recommend to people just starting out in mass spectrometry data annotation?
I would say the very first starter pack is to do that manually and to understand what you are doing, because it's a joke, but not really. Being able to give the right annotations to an ion, so find the right adduct we are looking at, the nature of the ion we are looking, and have a look to MSMS spectra manually, and be able to see if the quality is okay before going to databases, external or internal. It's a good start. But then, yes, any tool with a graphical user interface allowing you to see things before doing it automatically is my first guess.
Okay,
but then we need automatic tools to be able to process thousands of data.
Okay, thank you. We have 2 minutes for other questions. So we have a question from MP. How to assess quality of spectra, and are they commonly used indicators?
No, they are not commonly used indicators, not yet, but some tools are giving some scores to MSMS spectra. So we have MS purity, for example, in R, that is giving a purity score for the isolation of the parent ion, with a score between 0 and 1, 1 meaning that the parent was alone in the isolation window, so alone in the quadrupole before fragmentation, and 0 meaning that we have many ions in that, usually 1 Dalton or 0.7 Dalton isolation window of our quadrupoles. So that's one of example.
Okay. Raphaël Le Guen asks, can you speak about the difference in MS2 spectra generated by different machines, i.e. Orbitrap versus QTOF, and how it affects spectrum matching in GNPS or other?
Yes, being able to use filtered database is, I think, a good way to go, because we know as analysts that the spectra generated by different kind of fragmentation are different, even if we are looking at the same molecules. So it's possible now in a mass bank, for example, to filter and to keep only Orbitrap HTD hits or QTOF hits. So I would advise you to create sub-database for each of your instruments before running the the annotations, because sometimes in the huge aggregated databases we have a mix of GCEI, ESI, QTOF, ESI, Orbitrap, all together, positive and negative modes. Sometimes it's mixed, so it's really important to be able to create sub databases before starting the interrogation. That's my opinion.
Okay, and maybe the last one before switching to Melanie. Is there any new recommended tools and or libraries for GCEI HRMS from eClass?
Part of the community is now have access to EI-HRMS data, and so MSP files are available. I think of one in Resetox lab in Czech Republic that released something on Zenodo recently, and we have still the NIST database that contain some tools.
Okay, thank you, Yann. Melanie, are you here? Can you hear me?
Bye all. Thank you.
Thank you, Yann.
Hello, can you hear me?
Yes, I can hear you. So I've given you the right to share your screen. Yes, it's okay.
Yes.
So thank you, Melanie, to accept the invitation. So Melanie is a junior research group leader in MALDI imaging at the Institute of Surgical Pathology at the University of Medical Center at Freiburg in Germany, and the visiting scientist in the laboratory of Professor Olga Vitek at Northeastern University, Boston. She did a PhD in translational proteomics and worked as a postdoc in the group of Professor Olga Vitek on Cardinal, an open source software for mass spectrometry imaging data analysis. Our group is establishing open source tools and reproducible data analysis workflow in the Galaxy platform, and is working to improve the quality control, standardization and reproducibility of MALDI imaging to optimize its application to clinical tumor tissue. And Melanie going to present us pitfalls and open source solutions for mass spectrometry imaging data analysis. And the floor is yours, Melanie.
Thank you very much for the kind introduction, and also thank you for the opportunity to present some of our work here. So my junior research group has two different parts we are interested in, and one part is really the use of mass spectrometry imaging to analyze tumor samples, and the other part is to make mass spectrometry imaging more standardized in terms of wet lab, but also computation. And here I'm now talking about my postdoc work in the VTech lab, as well as some of our software things we did in Freiburg. So a short introduction, what is mass spectrometry imaging? You can think about it like a molecular microscope. You have a thin tissue section, and with mass spectrometry imaging, you can scan over the tissue section and obtain heat maps of different molecules directly from this tissue. And there are different approaches how this can be done, but overall one can say that many different endogenous and exogenous analytes can be measured, for example, proteins, peptides, glycans, metabolites, and lipids. And classically, it's done in a label-free manner in an explorative approach, but other approaches are also possible. Here are some examples of metabolite imaging studies published by other research groups. On the left-hand side, we see a classification study on thyroid cancers, and that's from the Elie lab. They had two types of thyroid cancer, one which was benign and one which was very aggressive, so very malignant. And on the level of the biopsy, which is taken here from the thyroid, it is not really easy to distinguish the two types, but of course they need very different treatment. And you can see that based on the metabolite heat maps, it's possible to get different distribution profiles. And based on this, they were able to distinguish the two types of thyroid cancers with more than 90% accuracy. On the right-hand side, we see that during maturation of a strawberry, different metabolites increase and decrease in different areas of the strawberry. So it can, of course, also be applied to study food, for example. Another area of small molecule imaging by mass spectrometry imaging is to image a drug. So the distribution of a drug can directly be measured when the drug is, of course, able to be ionized. And on the left-hand side is an example of a whole rat section, and olanzapine was administered and is distributed here, for example, in the brain and spinal cord. And then the metabolites of this drug are more located towards the liver and the bladder, meaning that they get secreted. On the right-hand side, we can see that we can also do a sort of targeted and more absolute quantification. So classically, it's all relative quantification, but it's possible to do a so-called mimetic tissue model where you have the one analyte, metabolite, or drug of interest, and you spike it into a tissue homogeneate at different concentration, and then you cut a thin section of this, put it next to your tissue of interest, and then you have here a concentration curve which also contains the same background tissue metabolites, and you can put a calibration curve, and with such a method it's possible for some selected molecules to also get an idea about the absolute quantity in the tissue. What's going on inside the mass spectrometer? Here's an example of a MALDI-TOF, which is probably the most used mass spectrometer. On the left-hand side, we see a glass slide with a thin tissue section mounted onto it and covered with a matrix, which is necessary for ionization. And this is put into a slide holder, which is inserted into the mass spectrometer. And there, when we do MALDI, we have the MALDI laser, and the MALDI laser scans over the tissue section and generates every few micrometer a full mass spectrum. The analytes are measured in the TOF tube. And then in the end, for every location where we were hitting with the laser, we obtain one spectrum, which in the end ends up with tens to hundreds of thousands of spectra, which we can put back together, because for each spectrum we know the location where it was generated, and then we obtain these heat maps with the different distributions for each metabolite or each analyte we're interested in. So here's an example of a pig fetus, and we see one mass is located in the heart, one in the liver, and one in the brain. And for many methods, it's possible to afterwards use the exactly same tissue section and do a staining, for example, here an H&E staining. And then during data analysis, the information can be transferred in both directions and gives much more insights into the morphology of the tissue, which helps to map the molecular content to different regions of the tissue. Nowadays, sometimes there's ion mobility in between here. And this, of course, helps us to get a certain separation of molecules and to obtain CCS values, which enable or help the annotation of the molecules. And as you can appreciate, typically we are just operating at MS1 level. It's not so easy to do MS2, even though some new studies showed that the field is moving towards MS2 imaging. But so far the standard is MS1, which of course also poses hurdles towards analyte annotation. So typical challenges in the field of mass spectrometry imaging data analysis are that we obtain many spectra with many peaks. So typically we have hundreds to thousands of mass over charge ratios. So we have many features. And as I said, we can have several 100,000 of spectra in one data set. So often a data set has hundreds of gigabytes. Then, because we are measuring the spatial dimension, it's a problem that the different spectra are not independent for statistical analysis, because we assume that spectra that are next to each other have very similar content. So there's a certain spatial dependency between them. And then we can use many different mass spectrometers and different ionization sources, and with them we can measure different analytes. So the spectra look also quite different and require different pre-processing methods. As I said, analyte annotation is not so easy because we don't have an LC in front. We only have MS1 level spectra typically. And that's also a major challenge in the field. What is typically done is that we do just an accurate mass matching to existing databases, or doing additional LC-HRMS analysis and matching these information to our MC features, or doing in situ MSMS. But all of these require, again, a lot of work and often manual work, and to improve the confidence in analyte annotation. There's also Metaspace available, which changed the field in giving us the opportunity to use our MS1 spectra and get an FDR calculation for matching to databases. So there's more confidence. It also allows to share raw data, and so you can upload your dataset to Metaspace and you get annotations. And you can also decide if you want to share your dataset with the community. And by this problem mitigated a bit. So now we have much more public datasets, but we hardly have any code and don't know how everyone is doing analysis, because the standard in the field are proprietary software. And many of these issues are to a certain degree solved by the Cardinal software. So the Cardinal software is developed by the VTech Lab, and the main developer is Kylie Bemis. And for my postdoctoral work, I joined the lab and worked on the version 3 of Cardinal. And Cardinal already has a very rich methods portfolio. It's in bioconductor, so it's R-based, it's free, it's open source. It's compatible with larger than memory data sets. And in the version 3, it's now better compatible with high mass resolution data set. It's even faster and more computationally efficient, and it has several new features. For example, better visualization, pre-processing, and new statistical methods. When we look at the typical data analysis steps, it hopefully starts with a quality control. Then we filter and combine our files. We do spectral pre-processing. Co-registration means that we overlay the optical image, for example, the H&E stained image, the purple thing I showed you, with the mass spectrometry imaging data. And then we do the statistical analysis and visualize our analytes of interest, and afterwards try to annotate as many as possible. And I want to now go deeper into statistics, because statistics is really the main focus of the Cardinal software. So Cardinal covers all of these steps except for annotation, where we provide a standard file format that is compatible with the typical databases or software that are used for this step. And the statistical portfolio is really the main focus of Cardinal. So what are typical analysis that we do? So one approach is to do an unsupervised analysis, like a segmentation, which means we don't provide any input. We are just asking the algorithm which of these spectrum is a pixel, so which of these pixels have similar molecular content, and then all the similar ones get one color and are one spectrum, are one cluster. And then for each of them, we obtain an average spectrum and also a ranking, which of the masses especially contributed to the clustering. And when we then plot the heat maps of these masses, we see that they correspond to the clusters that were found. The other way around, we can also do supervised analysis, which means that we are providing labels, information about which tissue is which condition. And here's the classic example of tumor tissues and normal tissues. And then we can train a classifier, and the classifier is then built in a way to predict for new samples if they are tumor or if they are normal tissue. What is special about the spatial shrunken centroids classifier, which is also available in Cardinal and also developed by Kylie Bemis, is that it really takes into account the spatial dependence of the pixels. And furthermore, it allows to shrink down the mass features that are used for the classification, so that in the end, only the most important mass features are actually included into the classification. And this helps to then get an idea which are the features that most contributed to my classification. And then it's easier to follow up on these features instead of following up on hundreds or even thousands of mass features. So to do the classification, we typically obtained very detailed annotation of pathologists, and that's quite a time-consuming task. And therefore, we developed an advanced classification algorithm, which can work with only rough annotations. So the idea is that, as you have seen in the picture before, we just say this is kidney cancer or it's healthy, but we don't say which part of the tissue is actually the cancer. And this saves a lot of time, because typically we do what you can see on the right-hand side. The pathologists are, so annotation in this context means that we are giving a label to a certain region of the tissue, and the pathologists here are really circling in really small areas where they say this is tumor, this is healthy, and so on. And we used such a detailed data set to show that the multiple instance learning convolutional neural network that we set up performed the best when we are only using the whole tissue label. And with this algorithm, it now saves a lot of time to pathologists, because we just need to know the tissue label, but we don't really need these very detailed annotations of the tissue. Further analysis available in Cardinal are just a classical class comparison. So here we do not care so much about prediction. If a new tissue comes in, what is it? It's just we have two classes and we want to compare the molecular content. And this approach uses linear models to find molecules that are changed. As we are in the spatial dimension, another approach that's interesting is co-localization. And this means we are putting in information about one feature that we like, and then we are asking which other features have a very similar distribution. And then we obtain correlations, and here the correlations are not so perfectly as we see in these images. So actually this 743 mass was the only feature that really had a distinct distribution in this part of the tissue. And similarly, instead of putting in a mass feature, we could also put in such an annotated region where we want to know which molecules are located in the region that is annotated. In addition to using Cardinal, my laboratory has also used Galaxy. And I guess many of you know Galaxy, but still I want to highlight that it's really great to deal with large data sets and to enable sharing raw data, but also the analysis code or the analysis that was done. So Galaxy is a web-based platform. It's also open source. And what is important is it has a graphical user interface. And it has already many thousands of tools from many different domains, also mass spectrometry, also imaging. It allows you to create workflows, run them on public clouds. Everything is reproducible and tracked, and it's super easy to share data. So we are mainly using the European Galaxy server because it's started in Freiburg, I know that Workflow for Metabolomics has even their own server, which is free to use. And what we did is we included the major Cardinal software into Galaxy because we thought it would be super handy to not just use Cardinal in R, because not everyone is familiar with R. And also we wanted to make use of this workflow engine that is behind Galaxy. So what we did is we wrapped very modular tools for all the different steps of mass spectrometry imaging data analysis. And in addition to Cardinal, we also used the R package multiquant and the Python scikit image for the overlay of the optical image and mass spectrometry imaging data. And so in Galaxy currently we cover all important steps. And what I would also like to highlight is that we generated an automated quality control tool, which provides an PDF report, which shows many data properties and gives an impression about the quality of the measurement and which pre-processing steps and parameters might be suitable. So all the tools are available on the European Galaxy server, but also in the toolshed, from where they can be installed easily into any other Galaxy instance. How's the timing? Oh, just a very quick outlook to finish up. So this is an example of peptide multi-imaging, but in the end, what I want to tell you is we had two types of cancers which we want to classify, and it doesn't really matter that it's now peptides. What is important is we could use these Galaxy workflows and generate 7 different Galaxy histories and share the raw data and the whole analysis history. And to our knowledge, this is really for the first time that mass spectrometry imaging of clinical tissue samples was made so transparent that everyone can look up every step of the data analysis and even reuse the raw data. So here are some links and resources in case you're interested to join the Galaxy Metabolomics. It's like community. So we are trying to set up Galaxy Metabolomics and proteomics communities. We have a chat channel and a mailing list, and sometimes organize also webinars on different topics, and you're very welcome to join these global efforts. And with this, I would like to thank my group and also the VTech Lab and all collaborators and you for your attention.
Yes, thank you, Melanie, for this nice presentation concerning metabolomics imaging. Is there some question? So you have time. We have time for small questions. I have just one question concerning the future of imaging, of metabolomics imaging. How do you see the future? So in diagnosis or in research?
So I'm located at the pathology department, and what we are really hoping for is that we can build classifier that can help the diagnosis of different, especially tumor types, so that our classifier based on the metabolomic profile is telling the pathologist that based on the profile, it's probably the subtype of cancer, because there are several types of cancer that are not so easy to distinguish for them just by looking through the microscope. And there are many studies that show that the molecular fingerprint is really helping to distinguish these tumors.
So you think you can implement that in the routine diagnosis? So you are searching for that?
Yes, so that's one of the plans, but until it's really allowed to be used for decision making, I think it's another 10 years.
Yeah, sure.
But that's the dream, yes.
It's not too time consuming for one analysis of tissue?
No, so it's very fast. And I mean, there are tissues in pathology that arrive while the patient is still in the surgeon room. And there you really need to get the feedback within 1 or 2 hours. And that might be maybe a little bit too fast for us. But most of the diagnosis is that the cancer is taken out, and then the pathologists also take up to 1 week to come up with a diagnosis.
Okay.
And that we can manage easily. But even the insertion is not completely out of scope. So there are some ideas and tricks how to make it fast enough.
Okay, if there are not other questions, so many thanks for this presentation, the answer.
Thank
Melanie.
you. Thank you again.
So we will continue the webinar with Nicolas Elie from the Institut de Chimie des Substances Naturelles from GIF-sur-Yvette. Nicolas is a research engineer at the CNRS, assigned since 2010 to the Institut de Chimie des Substances Naturelles à GIF-sur-Yvette. So he's particularly interested in the processing of mass spectrometry and is developing a new annotation software. So he will talk about a well-known software, MedGem, developed in collaboration with Marc Litaudan groups. So the floor is yours, Nicolas.
Hello, thanks, Pascal. So, as you said, I'm an engineer in the mass spectrometry team at ICSN, and I am the main developer of MedGem, a software dedicated to molecular networking since 2018. Okay, so what is a molecular network? To explain, we will look at this data set extracted from lipid blast. It's a collection of lipids in silico MSMS spectra. In this network, we can see nodes, which represent MSMS spectra, connected by edges, which are related to spectra similarities. To generate the network, spectra are compared 2 by 2. So each spectrum is treated as a vector in an n-dimensional space, 1 dimension for each peak. Then to compare two spectra, we calculate the cosinus of the angle between the two vectors. So if the spectra are very similar, vectors will be parallel and the cosinus will be close to 1. On the other hand, if the two spectra are very different, vectors will be orthogonal and cosinus will be close to 0. So the cosinus formula used here is slightly modified to take into account specificity of mass spectrometry. For example, these two DAGs differ by only 1 double bond and will contribute positively to the similarity score. But natural losses are also used in the calculation, and the peaks around 360 will contribute to the similarity score because they correspond to the same loss from precursor ion. Before comparing spectra, multiple filters are applied to the spectra. So the first one is the low mass filtering. So we just remove all peaks below a predefined M over Z value. Then parent filtering will remove peaks with M over Z in a range center on the precursor ion. The threshold filtering, we keep only peak higher than threshold. And then the window rank filtering. So the idea is to center a range with chosen width on the first peak of the spectrum. So if this peak is not one of the n highest peak in the range, we remove it, and then we continue with all the other peaks in the spectrum, one by one. So we end with this kind of spectrum. And the last step is to square root the intensity because highest peak will have a huge impact on the cosine score. In the end, multiple algorithms can be used to render the network. The classical one is a force-directed layout, which is used by GNPS or Cytoscape. And MedGem introduced another one based on t-SNE embedding. In a force-directed layout, nodes are modeled as charged particles that repel each other. And similarity between spectra are modeled as springs whose stiffness is proportional to the score. Nodes are first randomly placed, and the forces are calculated step by step until an equilibrium is reached. So here is an example of a force-directed layout applied without any processing step. So it is very dense and will be difficult to interpret. So to help a little bit, two mechanisms are used. First, a score threshold is defined, and all edges with a score below this value are cut. Then a top K mechanism is applied. This means that each node will have a maximum of K neighbors. An edge will be kept if and only if it is in the K highest of all the edges for both nodes it links. In this example, we'll use a top 2 mechanism. The two strongest edges of the blue nodes are highlighted in yellow. If we consider the strongest edges of the orange node highlighted in green, we can see that the edge between orange and blue will be cut because it is not in the two strongest edges for both nodes. So finally, we get this kind of network. We have clusters that we should be able to identify, but there is no information about distance between clusters, as they are just sorted by decreasing number of nodes. That's why t-SNE-based layout was introduced in MedGem. It's a non-linear projection of an n-dimensional space into a lower dimensional space with priority given to local distances over global distances. A quick explanation of the t-SNE algorithm. Let's consider this 2D dataset and try to embed it in 1D. So at first glance, some clusters can be seen. If we try to project this dataset to one dimension by using principal component analysis, we can see that the clusters are not well separated in the projection. t-SNE projection is better because nodes that are spatially close in the original space are close in the projection. And we can do a bit better by excluding nodes that don't have any neighbor from the projection. The result is this kind of visualization with nodes grouped into clusters with meaningful inter-cluster distances. And if we compare force-directed layouts and t-SNE layouts, we can see, for example, that these two clusters in yellow in the force-directed layout are spatially closed in the t-SNE projection. A few important features of MedGem. First, if you can add as many views as you want. They are linked together, meaning that nodes selected in one view will also be selected in other views. You can load a table of metadata associated to each node and interact with this table. Unlike GNPS, visualization parameters can be changed interactively, so you don't have to compute again the score matrix. You can draw a pie chart on top of nodes, for example, intensity of each ion in multiple extracts. And let's not forget the basics. We are working with mass spectrometry data, so we can visualize spectra associated to nodes, compare two spectra, and see which fragments and which natural losses contributed to the similarity score. We can filter nodes to find spectra with specific fragments, natural losses, or precursor. So in this example, we looked for phosphocholine fragments, leaving only PC and LPC nodes. And the last important feature is database query. You can find standards or analogs for specific nodes or whole network from 3 main sources with more than 3 million spectra available. So I would like to thank you for your attention. If you have any suggestion, need, or special features that would like to be implemented, feel free to contact me. Now I will be pleased to answer your question in English or French.
Thank you, Nicolas. Thank you very much for your presentation. Have we got some question in the chat, maybe? Not for now. Maybe it will be coming. So I have one maybe. You showed the similarity score on MedGem. On what criteria are the score based? It's between the M over Z. It's between the intensity of the peak.
We try to find a match between when you compare to spectra. We try to find spectra that are matching, meaning that they are with M over Z close to each other, and then we just multiply the intensity from one spectrum.
Okay, so M over Z and the intensity.
The intensity and the M over Z.
Okay, thank you. I think we don't have other questions, so please feel free to contact Nicolas if you have any question. And thank you very much, Nicolas, for your presentation.
Thank you.
So now for the last but not least presentation. Edern, I think you can share your screen now. Tell me if it's okay. Okay.
Is it okay?
Yes, but
you hear
not
me?
in full screen. Yeah, we can hear you. Not in full screen, but it's
It should be coming.
yes, it's okay.
Yeah.
So thank you, Edern, for accepting the invitation. So Edern is a research engineer in nuclear magnetic resonance at MetaBoA MetaTool platform, located in Toulouse Biotechnology Institute of Toulouse, France. He did his PhD in metabolomics and fluxomics in the MetaTool platform on the characterization of the photoautotrophic metabolism of the microalgae Chlamydomonas reinhardtii. I don't know if I pronounce good, using instantaneary 13C labeling flux analysis approaches. He was recruited in 2015 at CNRS and is since in charge within the NMR service of MetaTool Fluxomet platform to perform and develop new experimental and analytical techniques for metabolomics and fluxomics analysis. And the floor is yours, Edern.
Thanks a lot. Thanks for the introduction, Amandine, and thanks for the invitation. So to change a bit, I will speak about NMR. I think if there is any more people, please stay here. MS people too. So I will speak about C13 fluxomics, and especially so few developments that we did on the platform for a few years. So just a small introduction about flux analysis. The objective of a flux analysis, a complete one, is to get a flux map. What is actually a flux map is the distribution of intracellular carbon and energy fluxes in vivo, and it represents actually the actual the actual activity of metabolic networks in a specific context. We can differentiate two different approaches to get to exploit fluxes. The first one is to get an idea about the topology of the metabolic network. So is to try to identify the network topology, the network activity, and to try to find the structure of the metabolic networks of organisms. And we can also, thanks to this type of approaches, analyze compartmentation or channeling of enzymatic activities. This is what we are calling so metabolic network topology analysis, and it is more a relative quantification of fluxes. But we can also get, thanks to that, when we know a bit more about the topology, we can get a quantification of metabolic pathways, and this is what we are calling, in the end, the true fluxomics. And this is actually the objective of this, is to get a flux map and to measure carbon fluxes and energy fluxes in response to environmental or genetic modifications. Of course, it has a lot of applications in many fields, from biotechnologies to health researches. What you have to get in mind is that there is no direct method to measure intracellular fluxes, and so we need to go through indirect analytical approaches. The most common approach is to use C13 labeling, or in general labeling, but I would prefer one because we are focusing on carbon metabolism, is to use C13 labeling. We have the chance in nature to have only 1% of C13 labeling, so that means that we can modify the C13 composition of metabolites, and especially for to analyze a metabolic network is quite useful, because here, for example, you have an example at the left of two different really simplified pathways where we have different enzymatic reactions modifying A into D. And because we know that there is this difference in terms of carbon skeleton reorganization, we can, by analyzing the products of these different pathways, so in the end, the products, which is P, by analyzing the difference between the proportion of those two labeling forms, we can actually decipher and quantify the different proportion of the two pathways. Of course, if we have a more complex network, which is the case in life, this is much more complex, and we have to go into metabolic models. In the end, to get a flux map, we will need different needs, different tools. First, to get a model, a stoichiometric model of our metabolic network, central metabolic network, and carbon transitions, so to know how the carbon are reorganized between the metabolites in the network. We will need to measure what is going in and going out from the cells, so that means the extracellular fluxes, and it's mainly done with NMR. We will see that we also can measure the labeling of the carbon which are entering and going out from the cells, and we will need isotopic data that can come from NMR or from MS, or combined. Sorry. The first thing that we can do with NMR is to measure with a real quantitative manner and untargeted manner what is going out and coming in the cells by analyzing supernatant. It's a really simplified protocol with NMR, and we have the chance for that with NMR that is unrestricted and completely untargeted analysis, and we can get quite easily absolute quantification of metabolites. But we have also the chance to get, for example, in this case here, the labeling of compounds that are going out from the cells. Thanks to tools that were developed in the lab a few years ago, and that are still developed, we can get from those data really easily the specific rates of consumption and productions of the compounds outside, and so we get directly data that we can provide to the models to calculate fluxes. Okay, so how with NMR we can measure labeling? Here on the top we have so spectra, really simplified spectra from glutamate. At the left we have a proton spectrum, which is simplified, and we can get the different positions of protons. So thanks to NMR, we have also structural analysis, because we have redundancy of signals coming from the same molecule. And then, for example, we can focus on the proton 2 of glutamate, and we will be able to differentiate protons that are linked to C12 and protons that are linked to C13. And of course, if we have a mixture of both, we will have actually a spectrum which is the one below. I don't know if you see my pointer. I think not here.
We can see your mouse. Yes. Okay, it's a pointer.
Yeah. So in the end, if we have a mixture, we will be able to quantify the proportion of proton linked to C12 and linked to C13. At the left, at the right, sorry, we can also detect C13 directly in NMR, and we will have information about every carbons of the molecule. And if we are looking now directly on the C2 of glutamate, if we are pointing on it, we will be able. Sorry.
Et le conseil départemental, qui sont aussi des partenaires relativement incontournables.
I don't know speaking.
Yes, I will. Can you shut your micro?
Participe à la révolution numérique qu'on connaît tous et tous.
I will do it myself.
Okay.
If I can sound. It's okay, I think.
So we continue after the conseil départemental. So if we are looking at the C2 of glutamate, we will be able this time to measure the labeling of the neighbors. That means we will be able to have a different spectrum if the C1 is labeled, if the C3 is labeled, and if both are labeled. Of course, if we have a mixture of all these forms, we will have at the maximum a spectrum for this carbon of 9 peaks. This is what we are calling positional isotopomer, and in the first case, this is a specific enrichment. This is really the most detailed information in positional information that we can get with an analytical technique. And both combined are called cumomer, so for cumulated isotopomers. Of course, if we have all this information on the same one spectrum in 1D, 1D proton, for example, in this case, this is quite crowded and will be not. We won't be able to actually extract any information on labeling informations from this because it's too complex. So we will need to go through multidimensional NMR. And this is what we developed for many years now. And we developed different strategies from indirect, so going through protons. For example, here, this is an example of heterogenes, where we can extract protons linked to C12 and protons linked to C13, and this is a quantitative sequence. The same is doable with ZQF toxi, and we can quite really fast have informations on a crowded spectra that is not exploitable in 1D. We have also the possibility to use heteronuclear isotopic sequences to get positional informations. And for example, here in isotopic HSQC, we can extract positional isotopomers quite easily, and because we are respecting quantitative conditions, we can extract the different labeled form from them. So let's speak now about amino acids. One of the main targets that we are analyzing to get information about the central metabolism is amino acids. Of course, amino acids are quite easily to get because they are highly abundant in biomass composition, and we can extract them by hydrolysis of proteins. And thanks to that, we can have many informations about the central metabolites that are the precursors of those amino acids. And if we are able to measure the labeling of amino acids, we can come back to the labeling of the precursors in certain conditions. One idea was a few years ago to exploit those amino acids by a new way. That means by exploiting N15. That means exploiting nitrogen from these amino acids by labeling metabolites in N15. Why N15? Because N14 is actually not really detectable with NMR, and to use N15 can reach information on labeled form, labeled carbon that are not reachable with proton or direct carbon NMR. So we try to see if we were able to detect N15 in amino acids, and here we can see proton linked to NH3 form. So a proton linked to N15 in red and N14 in blue. we can clearly see that actually we have a small separation of a small split of the peak. That means that we have a correlation between protons and N15. So thanks to that, we can transfer magnetization from protons to N15 and go further, which we can see here is a correlation between N15 and protons. And so we have a map of the different position on N15 in the different mixture of amino acids. We developed, thanks to that, many sequences that are coming from the protein field at structural protein analysis at the beginning. And then we developed that to be able to see labeling and to measure labeling of free amino acids extracted from proteins. So I won't go into detail about all these information, but thanks to this approach, in these approaches, we are now able to quantify the labeling of the two first carbon of every amino acids. One of the things which is quite complicated to get is the C1 labeling of amino acids, and which is really rich of information into flux analysis. So as a matter of proof of concept, we were able to quantify the partition of glycolysis and pentose phosphate pathway on a series of mutants of E. coli of low expression or high expression of pentose phosphate pathway. So to come back on the complete workflow to get a flux map for one cell, it's quite can be quite complicated, especially if we have an organism that we don't know, and can be we need a lot of information, and we need time to process data and to be able to make the quantification and calculation of fluxes. So we try to go a bit faster, and we needed really to for that to get through automation and higher throughput. So in the platform for many years now, in collaboration also with the Metasys team in the lab, we developed a series of robotic platforms and tools to be able to automatize flux analysis from the cultivation, the sampling, sample preparation, and data analysis, data processing, and then finally to automatize and throughput the intracellular flux calculations. So we use for that combined analysis by LC-HRMS and NMR, and all the data coming from those two experiments were used to calculate fluxes in this type of experiment. We developed also a series of tools to increase the rate of analysis and processing of data. And in the end, a tool was developed also in the lab to increase throughput and increase also resolution of fluxes and precision. To exploit this workflow and to evaluate it, we decided to evaluate it on a series of mutants of E. coli collection, which is actually the KO collection, which is we decided to extract from those mutants a series of unknown functions mutants to try to see if from those we could see silent mutations that were affecting flux maps. And so we did in the end, in few months, not far from 200 cultivations and flux maps. And in the end, we got a huge amount of data from NMR and NMS that were exploited to make those flux analyses. From those 180 strains, actually two on these huge data sets showed really interesting fluxotype or fluxome, and actually we were able to see from a repetition of those strain cultivations that two mutants, YDSS and YBGP, had really clear different phenotype from the mass. Unfortunately, this was not sufficient for to direct link those mutants and those genes to a function, but it enriched actually the knowledge on this, and this is something which is still to do to go deeper into the analysis of those strains. So to conclude, just a word to say that I hope I show that actually we have a really powerful tool to analyze positional isotopic incorporation, which is really the most sensitive and the most rich of information tool to do so, which is decoupled with, which is combined with MS isotopic profiling. We developed N15 labeling also, which increase actually the coverage of isotopic quantification that we can get with NMR. And we developed also in the platform MS profiling, which is also exploiting N15. The workflow is in continuous development and improvement. We are still developing into the platform new tools and improving the tools that we have to get a fully integrated fluxomic workflow. And the more and more we are using the W4M platform in Galaxy to integrate this. And we still are developing also processing tools to go a bit faster into the processing, which is still the limiting step, because we have a lot of information and it's still a highly manual processing step. And we are developing also a few other labeling application and quantification also by using phosphorus labeling. And we are going to develop really soon a dynamic monitoring also of metabolic fluxes with the acquisition, I hope soon, of microfluidic NMR probe to come. I would like to thanks everyone, and if you have any questions, if you are interested into these tools, don't hesitate to ask and to contact us. Thank you.
Okay, thank you, Edern, for this nice presentation. This time's about NMR, but not only. I think there's a question from Gina Ajar, if you are ready. So the question is, you mentioned that it took 3 hours acquisition for an HSQC to get a good resolution. Is it plain or possible to test recent approaches in 2D NMR, so such as NUS or spectral aliasing, to have a faster acquisition?
Yes, so thanks for this question. Yes, actually this is possible and it is planned. We actually are in MetaBweb. We have a project to establish the good conditions of NUS on every platform that are actually equipped with NMR in MetaBweb. And this is a program which is planned, which will be continued in this year. The thing is that we really need to get a really high resolution in F1, so in the indirect dimension for HSUC. And this is a bit the limiting step if we want to really be able to separate the different isotopomers, because they are quite well separated in chemical shift in the second dimension. But the pattern of the labeling, the different labeling form, is actually measured on few hertz. So we really need to try NUS, but really we don't want to lose resolution on it. And this is one thing we have in mind to evaluate this, the ratio of NUS that we can use.
So, but you know that with NUS you can also increase the resolution. Instead of a gain of time, you can also reduce the observation windows, but increase also the resolution. So
yeah, yeah, this is something we have to try because of that, and to not lose because this is always a matter of sensitivity and resolution. So I think that for those type of samples, for amino acids, for example, we are not really limited. So it should be, and especially if we are labeling the C13s. So yes, this is in the tubes, as we say.
okay. Is there other questions? If not, I have just one question. So
Yeah.
you're just working with bacteria, so strains, which, if I'm right, are suspension cells in suspension. Yes.
Yes.
Or,
Yes. Yeah,
do you plan to work with eukaryote cells or human cells, for example, or animal cells,
yeah.
or is it a difficulty?
Yeah. Actually, we are not working only with bacteria. So this is, of course, the specialty of the lab where we are. And to develop tools for the flux analysis, we are, of course, using microorganisms because it is more simple and easy to grow. But we are working with many other organisms, especially and including animals, animal cells in cultivations, adherent cells in cultivations, when we are also performing labeling on them. And we have the tools we are developing analytically are actually the same for those cells. The complexity to get fluxes on those type of cells is actually that we have there is two main difficulties. Is that they are growing complex media, so we need to to find the good label substrate to get the right information we want, or to repeat the labeling experiment with different carbon sources to get a resolution of the fluxes, because all the other compounds that are consumed will dilute the labeling information, and we lose a lot of information from them. And also the other complexity is the compartmentations of those organisms, which complicate complexify, of course, the analysis, because we will have also a mean of labeling of, for example, the same metabolites in different compartments. So we lose also again resolution. So we need to have the good models and actually the good tools to do so. But this is an ongoing project into MetaBweb, and if you have any question about that, I can speak a bit longer on this. And I have a lot of colleagues that I can also show you what we are developing here in terms of compartmentation and co-cultivations of organisms. So yes, there is a lot of things that are quite exciting to ongoing.
Okay, many thanks again to Edern for this presentation. And
You're welcome.
okay,
Thanks
I want to also to thanks all the participants, all the speakers to this webinar, and you, all the people that have followed this webinar with a high peak of participants, so which is quite nice, and we more than 180 participants at one point. So it's quite nice for the webinar, and I hope that the future webinar will be also followed by a lot of people. So thanks again for your participation. So now we have some information for the future. Amandine?
Yes, thank you for all the speakers today. So just a quick event upcoming from RFMF. So you have the European School of Metabolomics, who takes place in Granada, in Spain in April 2024. So you can subscribe on the link, and Yann talked about the scientific days of the RFMF, which take place in Saint-Malo the first week of June, and you can subscribe very soon to come to this event. And the next webinar will take place in March the 14th. So we will distribute very soon the flyer with all the speakers and dates for the next year. So stay tuned and see you soon. And thank you for today.
Okay, so bye and see you next year. Thank you very much.
Bye, everyone.